Hi everyone. Today we're going to cover some of the events that occurred during the first 2 weeks of development. I discussed gametogenesis in a previous video, and I'll review the important steps in fertilization here and show some examples of what happens when fertilization doesn't occur normally. I'm Dr. Catherine Moore, the Histology Wizard. Human fertilization is the union of a human egg and a sperm, usually occurring in the ampulla of the fallopian tube, also called the oviduct. We know that fertilization involves a series of highly coordinated molecular events that begins with contact between the sperm and the oocyte, and it ends with intermingling of paternal and maternal chromosomes during the first mitotic division of the zygote. And the overall result of this union is the production of a zygote cell, or fertilized egg, and the initiation of prenatal development. Now, before I review the steps of fertilization, I want to think about what the process has to accomplish, because this might help you better understand the reasons behind each step. First, the process ensures that the ovum is fertilized only by sperm of the same species, that only one sperm fertilizes one ovum, and that defective sperm can't succeed. And to accomplish all of these things, there are multiple behavioral and molecular barriers to fertilization. It's also important that factors in the ovum become activated to support further development. And finally, of course, the process has to ensure that the diploid state is restored in the zygote. So we're going to talk briefly about the process, and I'm not going to go into any great molecular details here, but I'll cover the basics and touch on some of the most important barriers that helps ensure that these criteria are met. So we'll start as the sperm are going to make their way through the female reproductive tract. This is the first step, sperm preparation or capacitation. Sperm undergo their final steps in maturation in the human female genital tract. And this is a biochemical modification of the sperm cell membrane, and it's required to make sperm competent to fertilize an oocyte. Once they reach the egg, sperm will use their flagellar tails and enzymes to drive through the corona radiata. And this is the layers of follicular cells that surround the oocyte. Now I'm just showing one sperm here, but actually multiple sperm can penetrate the corona layer. However, only capacitated sperm can bind to and penetrate the next layer, which is the zona pellucida, seen here in blue. Sperm penetration requires hydrolytic enzymes so that it can burrow through the zona pellucida. And this is the acrosome reaction. Now that red haze you see here represents the enzyme stored in the sperm head and then released. Now once a sperm penetrates the zona pellucida, what keeps other sperm out? Well, again, once that sperm hits the oocyte, this triggers the release of cortical enzymes from granules inside the oocyte, which I've colored here in green. So this is the zona or cortical reaction, and these lysosomal proteins are species-specific, and they permanently alter that zona pellucida, so they form almost like a shield or a barrier. And this step is critical to prevent polyspermy. Next, the oocyte membrane and the sperm plasma membrane will fuse. The head and the tail of the sperm will enter the oocyte, but importantly, the sperm plasma membrane is going to remain behind. Now, this step also triggers the resumption of the second meiotic division of the oocyte, and then the haploid nuclei will come together to form the zygote. Now, let's review the end results of fertilization. So again, the sperm head is going to enter the oocyte, triggering that second meiotic division, and then the pronuclear membranes will break down. The pronuclei will fuse, DNA replication will occur, and then we have the formation of the zygote. Now this process is going to ensure biparental inheritance and is going to determine the genotypic sex of the new individual. Now both gametogenesis and fertilization contain steps to ensure genetic variation. This is through independent segregation of chromosomes during meiosis and recombination of genetic material, so crossing over, in the gametes, and the mingling of paternal and maternal chromosomes during fertilization. So now we have a zygote, and what happens next? Well, we know that the zygote has to get from the oviduct or the fallopian tube to the uterus, where it can implant and then grow. So let's follow the zygote from fertilization, or day 0, to about day 7. So this cartoon shows the events that take place in the pre-implantation period. I've labeled each stage with its timing. Bear in mind that this timing can be slightly variable. So recall that the oocyte is expelled from the ovary during ovulation, and it's captured by the fimbriae, those finger-like extensions of the oviduct or fallopian tube. We know that fertilization will normally take place within the oviduct, and the resulting zygote will be swept along the tube to the uterus for implantation. Now, once the zygote is formed, it starts to divide, and it undergoes a series of mitotic cell divisions or cleavages without change in overall shape or size. All the while, it's moving towards the uterus. Now, this cleavage stage can last for about 4 days. Once the embryo reaches 16 to 32 cells, it gets a new name. We now call this the morula, which actually just means mulberry. At this early stage in post-fertilization development, the cells have now rapidly divided via mitosis, and the embryo is a solid mass that's going to give rise to the embryo proper, the extraembryonic membranes, and the placenta. Now, at about day 5, the morula develops a fluid-filled cavity. It's called the blastocele, and it's also given another new name. Now we call this the blastocyst. This thin-walled hollow structure contains a cluster of cells called the embryoblast, or inner cell mass, that will give rise to the embryo. Finally, between 6 and 7 days, the blastocyst will hatch from the zona pellucida, implant into the wall of the uterus, also called the endometrium. So now let's take a look at what this would look like under a microscope. These images are from in vitro fertilized embryos, and here you can see some of the cleavage stages, and then the morula, which will undergo a change in cellular properties, and this is called compaction. So essentially, the morula is going to compress into a tight ball. And then in around 5 days post-conception, we're going to see the early blastocyst. So you can see the cavity or blastocele, but you also can see the first 2 distinct tissues, the embryoblast, or the inner cell mass, and the trophoblast. And the embryoblast is going to go on to make the embryo, and it's the structure from which embryonic stem cells are derived. And the trophoblast is going to go on to make the placenta. Now, importantly, interactions between the embryoblast and trophoblast are absolutely required for subsequent development. If they're separated from one another, or if either cell type is prevented from coming into existence by, say, genetic mutation, usually you don't get an embryo, only a mass of tumor cells. So even at these early stages in the blastocyst, 7 days of development, there are distinct tissues, and interactions between these tissues are required for organismal development and function. Now I want to take a quick detour and talk about stem cells. Recall that stem cells have the potential to become many types of cells, and while they don't divide often, they can self-renew. And they give rise to cells that have more restricted potential, such as blood cells or hepatocytes or neurons, as I've shown here. Now keep in mind that embryonic stem cells are pluripotent. That means they can give rise to everything but the placenta. So they're not totipotent, which would mean they could also make placenta. Now the more restricted stem cells are lineage restricted. And this just means a hematopoietic stem cell can give rise to multiple types of blood cells, but it can't, for example, give rise to neuronal lineages. And in the embryo, once the embryoblast and the trophoblast form, these tissues, while they're still stem-like, are now more fate-restricted. So embryonic stem cells are isolated from the embryoblast, and this means that they are pluripotent. They can self-renew by dividing, and they can differentiate into any specialized cell in the body, but they can't differentiate into extraembryonic tissues, such as the placenta. Unfortunately, this also means that they can give rise to teratomas, tumors that contain many kinds of differentiated tissues. Now, based upon what you've just learned, before what embryonic stage would cells be totipotent? Meaning, when could they give rise to both the embryo and placental tissues? Well, if you said morula, you'd be correct, because it's at the blastocyst stage that we begin to see the first lineage restriction. Remember those first 2 distinct tissue types. Now, why did I take this detour? Well, it's because understanding what pluripotent and multipotent and totipotent mean and their relationship in the early embryo can help us understand how we currently think about the process of twinning. The easiest type of twinning to understand and explain is dizygotic twinning. Now here, oocytes are fertilized by two separate sperm, and each develops into a separate blastocyst, and the result is non-identical twins, each with their own placenta, chorion, and amniotic membranes. Monozygotic twinning is the result of a single fertilization event with a later division of tissue. Now, in the majority of cases, this results in identical twins. But whether those embryos share a placenta or amniotic or chorionic membrane is more complicated, and the result really depends on the timing of the splitting. So I'm going to way oversimplify this process to illustrate different times of splitting. Bear in mind, this is only one mechanistic model, and it probably doesn't explain all the different types of twins. The first type of twinning that I want to talk about is where twins each have their own placentas, chorions, and own amniotic sacs. So what this means is a zygote splits early, likely at 3 to 4 days during cleavage stages, probably at the morula stage. Now each one of these will develop into a blastocyst, and at hatching there will be twin blastocysts that implant. Let's look next at monochorionic diamniotic twins. Now here, the zygote is going to split at the early blastocyst stage, before or very close to hatching. In this case, there are two embryoblasts, but they share the same trophoblast, so they share a chorion and usually share a placenta. But they have two amnions because the amnion comes from the embryoblast, and at this stage, it hasn't started to develop. A final example is a case where the twins share a placenta, chorion, and amniotic sac. To get this result, a single blastocyst implants, and the embryoblast splits after implantation, sometimes right up to gastrulation. So at day 6, we have a blastocyst with its embryoblast and trophoblast. It's traveled down the oviduct to the uterus, it's hatched from the zona pellucida, and now it's ready to implant in the uterine endometrium, or decidua, where it can grow and be nourished. So let's review how that happens. So at about 6 to 7 days, implantation begins. Here's our blastocyst with the outer trophoblast in red and the inner embryoblast in blue. And we'll use this cartoon to see what happens next. First, the trophoblast is going to proliferate and split into two distinct tissues. The syncytiotrophioblast, shown here in pale green, and this is the inner layer of multinucleated cells that lacks intracellular boundaries, so it's actually a syncytium. Now this tissue will undergo epithelial to mesenchymal transition, it will produce enzymes including matrix metalloproteases that will chew up the tissue, and those syncytiotrophioblast cells will invade through the epithelium into the connective tissue of the endometrium or decidua. And this tissue is also going to produce human chorionic gonadotrophin, or HCG. And this hormone will maintain the corpus luteum of the ovary. And in turn, that structure is going to produce hormones that maintain the uterine lining and keep the pregnancy intact. The inner layer of the trophoblast is called the cytotrophoblast, and this tissue lies external to the wall of the blastocyst. These cells are different. They're irregular, ovoid shaped, and mitotically active, and they're critical for forming the fetal placenta and membranes. The embryoblast will also divide at this point, forming the epiblast, shown here in blue, and this tissue is going to give rise to the embryo proper, that is, all the tissues of the embryo. While the hypoblast, in yellow, is a more transient tissue that will contribute to the formation of the extraembryonic mesoderm and the yolk sac, and that in turn will give rise to the chorion. A few days later, the embryo has fully implanted into the uterine endometrium, and this cartoon shows it fully invaded. Now note the syncytiotrophioblast, with the beginnings of the trophoblast lacunae that will eventually develop into the intervillous spaces of the placental circulation, that cytotrophoblast, and the hypoblast cells now face and partially line the extracoelomic cavity or the primitive yolk sac, while the epiblast cells are going to form the embryo and the amniotic cavity, which will be surrounded by the amniotic membrane. And the amniotic cavity, of course, will eventually surround the fetus. At this stage, a little over a week of development, and of course because embryologists like to name things, we call this the embryoblast. We call the embryoblast the bilaminar disc because now it has 2 layers. Well, I've just gone over a lot of steps pretty quickly, and you can see that this is a fairly complex process. Now, what happens when events in this first 10 days or so go awry? Well, we know that usually disruptions to these very early processes results in the death of the embryo and spontaneous abortion. And one potential outcome of an abnormal fertilization process is called a molar pregnancy, which is essentially a pregnancy without an embryo. So a complete hydatidiform mole forms when an oocyte is fertilized normally by a single sperm, but then the female pronucleus is lost by some unknown process. And that single male pronucleus is going to divide to form 2 haploid nuclei, and those will eventually fuse to form a diploid structure. Less commonly, an oocyte is fertilized by 2 sperm, and the female pronucleus is lost, and a diploid structure is formed. In both of these cases, without the female pronucleus to drive development, there will not be an embryoblast and there will not be a fetus. There will only be trophoblastic tissue that becomes placental membranes. Now, in the case of a complete mole, all the chromosomes are from the father. And so what does this actually look like? Well, there's abnormal growth of the placental tissues coming from the trophoblast, and that's going to form basically a tumor that has a very characteristic grape-like appearance with no fetal tissue. This tissue can secrete high levels of HCG, or human chorionic gonadotrophin, and sometimes this causes the molar pregnancy to appear normal, but often the levels are too high for the suspected fetal age. Now, abnormal HCG levels may indicate a molar pregnancy, but nowadays most are detected early by routine ultrasound where the absence of an embryo can be seen. These moles can cause bleeding and nausea and will usually miscarry, if not detected earlier, by 16 to 18 weeks of gestational age. Now, moles are essentially tumors, and they need to be removed, as very rarely these benign tumors will give rise to metastatic carcinoma. A second type of molar pregnancy results in the incomplete or partial mole. Here, an oocyte is fertilized by 2 sperm, or by an abnormal diploid sperm. And what this results in is a triploid karyotype with 2 sets of paternal chromosomes and 1 set of maternal chromosomes. Now, because of the presence of the female pronucleus or chromosomes, there's usually some evidence of embryonic development. Now, these patients may have milder symptoms and the embryo may develop further along, but it's always grossly abnormal and is never viable. Now, these molar pregnancies support the hypothesis that the female genome directs embryonic development while the male directs placental development. Molar pregnancies are more common at extremes of maternal age, so people older than 35 or younger than 20, for reasons that we don't completely understand. Also, previous molar pregnancy, low-fat diet and lack of beta-carotene, a prior history of spontaneous abortion, or sometimes oral contraception. We know that the risk of a molar pregnancy is less than 1%, but unfortunately there can be other errors in the fertilization process, including ectopic pregnancy. Now, normally the blastocyst is going to implant into the endometrium of the uterine wall, kind of where I've drawn it here. However, there are times when the blastocyst will implant in an abnormal site, and this can be due to infection or inflammation of the oviduct or scar tissue. Sometimes the tube is formed abnormally during development or lacks the capacity to help move the oocyte to the uterus. The most common place for an ectopic pregnancy is in the uterine tube or oviduct in the region called the ampulla, which is where fertilization takes place most often. So 90% of ectopic pregnancies arise in the ampulla, and that's why ectopic pregnancies are often called tubal pregnancies. However, there are a number of other ectopic locations, for example, in other sites along the oviduct, including the isthmus or the fimbriae, near the opening of the oviducts into the uterus. More rarely, the blastocyst can develop deep in the uterine wall. This is called an intramural pregnancy, or in the cervix, the ovary, or even in the abdominal or peritoneal cavity. Now, the fertilized egg cannot survive outside the uterus, and if left to grow, again, depending on where it is, it can damage nearby organs and cause life-threatening loss of blood. For these reasons, an ectopic pregnancy is often considered a medical emergency. Now, to finish up, where are we at the end of week 2? Well, we're at the bilaminar stage. Now, as seen in the cartoon on the left and the colored micrograph on the right, we now have differentiation of the embryoblast. We have dorsal cells, the epiblast, that have organized to form the amniotic cavity, and those are going to give rise to all the tissues of the embryo. We also have ventral cells, the hypoblast cells, that are going to form the roof of the secondary yolk sac. Now, you may have learned previously or read older embryology books that state that the hypoblast cells are going to form endoderm tissue. But we know now with modern molecular techniques, including lineage tracing, that that's not true. We know now that all embryonic tissues in the body arise from the epiblast. Now, finally, it's at this very early stage that the embryo begins to organize along an axis. In this case, the dorsal side is composed of the epiblast, and the ventral side is the hypoblast. We'll see that the anterior, posterior, and medial lateral axes will be organizing during gastrulation. Now, I realize this was a pretty quick trip through some incredibly critical processes, and I know that early development is complicated, especially because the names of the structures change a lot in the first couple of weeks, but hang in there. Gastrulation's up next, where this bilaminar embryo will become a trilaminar one, and we'll see the formation of ectoderm, mesoderm, and endoderm. Be sure to check out my videos on gametogenesis and gastrulation. Thanks for stopping by.